Good evening, everyone. It's really great to welcome you. My name is Alexander Hoffmann. I'm the director of the Institute for Quantitative and Computational Biosciences at UCLA. I wanted to mention a few housekeeping items before we begin. This event is being recorded. In addition, the chat function has been disabled, but you can submit questions during the webinar using the Q&A function. As many of you appreciate, big data and computation have profoundly affected all aspects of our lives, and also all academic disciplines. In the biomedical sciences, this development amounts to what can be described really as a revolution. Whereas 20 years ago, computational work occupied maybe 5% of biomedical research effort, it is now more than 50%. That represents a radical change that affects not only how we do research, but who is doing biomedical research and what training we must provide and what we teach undergraduates interested in biomedicine. To guide this massive transformation at UCLA, I was tasked in 2014 to establish the Institute for Quantitative and Computational Biosciences. The Institute is a partnership between UCLA Life Sciences, Physical Sciences, the Health Sciences, and Engineering. It involves faculty, postdocs, graduate students, undergraduates from more than 15 departments, ranging from microbiology and immunology, which is my own department, to physics, to computer science, in engineering, and pathology in a medical school. We research a multitude of biological and biomedical questions. What unifies us is the mission to expand quantitative and computational biosciences research, training, and education. I invite you to learn more about QCB on our website and look forward to highlighting the incredible research and wonderful accomplishments of our faculty and students in future webinars. Today, I welcome you to our first webinar, Mapping the Brain with 100 billion cells. Is this an impossible task? Our brain depends on a network of specialized neurons that occupy specific locations and make specific connections to navigate life and make decisions. Neurons in the brains are specialized, and there are likely tens of thousands of different types of neurons, each type making different connection patterns and having specialized information processing capabilities. Before we better understand brain functions, we need a brain map that shows us what types of cells occupy different locations in the brain. This is an enormously challenging task, as there are thought to be close to 100 billion cells in the human brain. This number of 100 billion is in fact orders of magnitude higher than the number of nucleotides in the human genome. You may recall that the first map of the human genome was presented in 2002 after a decade-long international effort. And having that map transformed biomedical research and gave rise to the notion of precision or personalized medicine. Our three speakers today are undaunted by the complexity of the task to map the human brain, thanks to some major technological and computational innovations that they themselves are pioneering. And it will be a great pleasure to introduce them and then manage the Q&A for them. So first, I'm pleased to introduce to you our first speaker, Dr. Hongwei Dong. Dr. Dong is a professor in the Department of Neurobiology and director of the Brain Research and Artificial Intelligence Nexus, B.R.A.I.N, BRAIN, at UCLA. Previously, Dr. Dong constructed the original Allen Reference Atlas, which serves as the backbone to current genomic Allen Brain Atlas. Then Dr. Dong founded the Mouse Connectome Project, a pioneering large-scale brain mapping project that aims to construct a three-dimensional Google Earth-like digital connectome atlas of the mouse brain. The Mouse Connectome is an important research resource that's open to all neuroscientists worldwide. Dr. Dong is particularly interested in how the cerebral hemisphere regulates, coordinates, and synchronizes motor activities in goal-directed behavior associated with motivation and emotion. With his international collaborators, He's had a series of publications in journals like Cell, Nature, Nature Neuroscience, Neuron, PNAS. So please join me in welcoming Dr. Hongwei Dong.
Thank you very much, Alex. Very pleasure to be here. Let me share my screen. All right, so can everyone see my screen? Okay, so my title is Mapping the Brain, Construct the Whole Brain Wiring Diagram. So I would like to start some basic information here. Our central nervous system, include brain and spinal cord, receive and process information from outside environment, and process the information and then send this information to our body to coordinate motor behavior. And in the brain, fundamental functional unit is called a neuron. Each individual neuron receive the information and pass this information to their downstream other neurons. A group of neurons, they can work together organized as neural circuitry, and multiple neural circuitries, they organized as sub-networks. And so basically our entire brain is organized as these multiple different neural networks. Each individual neural network, they have specific function, process specific neurobiological signals to control our cognition, memory, attention, emotion, and other motor actions. And obviously human brain, as what Alex has mentioned, is enormously complex. It has 86 billion neurons, close to 100 million neurons, and 125 trillion connections. So basically, the study human brain is a tremendously big task. Fortunately, as a neuroscientist, we can study a mouse brain that is much smaller with 70 million neurons, a trillion connections. Importantly, use mouse brain, we can apply many advanced technologies like genetic technologies and other brain mapping tools, make it accessible, make it feasible to study the whole brain connections and their other functions. And also it's important to mention here, mouse and human shares 95% genome, and the vast majority of their neural connections, they maintain the similar organizational principles. So basically, it's about 10 years ago, NIH launched this Human Connectome project and also Mouse Connectome project with the same general objective, that is to map the whole brain, understand how the brain structures, they are organized as different functional networks. So here's just one example to show the neural pathways. We use the fluorescent dyes to label these neural pathways, connect one brain region to other different brain regions. Use different colors, we can label different neural pathways. So in this case, we go structure by structure across the entire brain. In the mouse brain has more than 1,000 different brain structures. Eventually, what we want to do is we map each individual brain structures, their neural connections, go structure by structure, eventually the whole brain. This is just like we construct a Google brain of the mouse. And this information will provide tremendous knowledge for us to understand how human brain works. And so what means the neural networks? And actually, in current field, people has been very familiar with this called artificial neural network. The artificial neural network actually is based on the relatively simpler real neural network. This each dots is called nodes, and they connect with each other through these connections called edges. in the artificial neural networks usually has this input layer, which means these artificial neurons receive the information, pass the information through other neurons in, it's called hidden layers. Eventually, all processed information sent it out through this called output layer. And in compare with these artificial neural networks, our real neural network actually is much, much more complex. And what I'm showing here, that's called cortical basal ganglia thalamic neural networks that was constructed based on the real data produced from my lab. So if we turn this neural network like this way, and that will be similar, it appears similar organization with artificial neural networks, but it's much more complex. This entire on the left row, that is all different cortical areas. They can be considered as an input layer. And on your right side, that's called a substantia nigra. That is can be considered output layer. But in the middle, these hidden layers, you can see how the cortical information passed to this in the middle. This is the first hidden layer called stratum, how the information convergent, divergent, and pass to eventually go to this nigra, this output layer. It can send the information back through the thalamus, back to the cortex. So basically, real neural networks is much more complex than the artificial neural networks, what we are working on now. And don't forget what I showed that this cortical basal ganglia, just one subset of the entire neural network for the cortex control entire motor system. You can, what I showed here is in each other subset of the either brain region or subset of neural networks, they have their own complexities. And also this networks only for control our motor behavior. Don't forget we have other system for control our cognitive functions. Here's a hippocampal wiring diagram. Hippocampus that is well known for our spatial memory, and also amygdala, that is another system for control the emotion. So we go system by system. Eventually, we want to construct the whole brain, they organized as neural networks. So basically, we want to put together, this is a whole brain wiring diagram. And so if we look at each individual brain structures, like in wiring diagram, it's just like one nodes. But however, don't forget each individual brain structure, actually they include hundreds of thousands of neurons as what I showed here. This is real data. We label the neurons in the amygdala as what I mentioned, that is structure involved the emotion like a fear. You see each individual neuron, they have their complex process, this dendrites. And so which means each individual neurons, they can process enormous information. But all these neurons together, they tangle together, this is just like a big jungle. So basically, how can we understand them? So we use different ways to label subset of neurons, as on the left side is video showed is green colors neurons. You can see this each individual neuron, they have their dendrites. Dendrites has these small dots that's called dendritic spines that receive the information. And this pink color process that is called axons. They come from another brain region, project to this green color region, send the information here for the information process. So basically, we use this way to label the different brain structures, label their input, output. Eventually, we want to understand the whole brain, how they worked. So if we look at this amazing video, this all real data, look at the individual neurons, they are just really complex, really beautiful. And a green color, that is axon, come from different brain structures. The pink is the neuron in the local. And so what we're trying to understand how brain works. When I talk to my student, I usually tell my student how brain works is just like a piano. Because one piano, they have fixed keys. One piano has about 82 keys, but with different combinations, you can play infinitive music piece. So our brain, even we have this, like a mouse brain has 100 million neurons, human brain obviously has 100 billion neurons. That is even more complex. But with the different combinations, they can conduct infinitive brain behavior. And so the organized, how these neurons, they are organized, that is through the neural circuitry. So our job as a neuroscientists, we're just really trying to understand these neural circuitries, how they organized. They put together different populations of neurons, different networks for conduct this enormously complex brain behavior and functions. And obviously, we map the brain not only just for fitting to the curiosity of our humans, but also this mapping the brain really help us, has enormous, tremendous applications for us to understand how brain works, understand the underlying mechanisms of the neurological neuropsychiatry disease. I just give one example. This is Alzheimer's disease. This is devastating to the whole world. And it's the AD called the disconnection syndrome. So basically, so we can apply our technologies to systematically characterize earlier and late stages, how the connectivity get disrupted. And hopefully this information can provide to our clinic scientists, clinic studies to understand that for the patients, to help the diagnostic, prognostic, and eventually to identify, to explore novel technologies for improve not only diagnosis, but also treatment and management of this, not only AD, but also many other neurological disease. So basically, I show you here, that is a mouse hippocampus labeled this individual neurons. You see this each individual neuron, just like what I already showed the videos, and they got a really tremendous complex morphology. We hope we label these neurons not only just in the look at the normal brains, but also look in the disease brains to look at what happened in different disease stages, how their morphology changes, what is the underlying molecular changes. That will help us to identify novel drug targets, eventually give us like novel ways for treat this disease. And so basically, in summary, we study the human brain. The history already is over several thousand years. And in the last 2 or 3 decades, we made tremendous progress. But however, until we really map the detailed brain circuitries, generally the whole brain wiring diagram, that potential is really huge. I don't want to see too much, you can listen other speakers, we can discuss this great question. I can stop here.
Oh, thank you so much, Dr. Dong. That was amazing. Those microscopy images and the videos really highlighted just the complexity and how much is already known, but how much we still have to go. So that's wonderful. Thank you so much. And we'll call on you for question and answers later on. Now it is my pleasure to introduce Dr. Roy Wollman. Dr. Wollman is a professor in the departments of integrated biology and physiology, and also in chemistry and biochemistry. He's also faculty in the Institute for Quantitative and Computational Biosciences, and my colleague down the hall. Prior to joining UCLA, Roy was an assistant professor at UCSD. He did his postdoctoral training at Stanford and received his Ph.D. at UC Davis. The Wollman Lab is all about developing and implementing imaging-based single-cell technologies and is applying them now to the brain. Please join me in welcoming Dr. Roy Wollman.
Thank you very much, Alex. Let me also share my screen. Excellent. So it was wonderful that Alex already mentioned the sort of a human genome project, because that's really where I also wanted to start. In 1990, I was in middle school, and the human genome project launched. And at the time, they were trying to think, how will we solve that? It was an enormous mapping puzzle, and they used the technology they had at the time. So really, I won't go into detail, but the gist was to take the genome, can take small chunks, put them in bacteria, amplify them, and sort of chunk by chunk, slowly map the entire sort of process. And that was a really big undertaking. And then somewhere along the way, there was a new technology, whole genome shotgun assembly, right? That technology all of a sudden changed the game. It allowed you to amplify the whole genome at once and map lots of small segments and just computationally kind of jigsaw puzzle-like, bring them all together to create really those sort of the full human genome project. And I was in college when the human genome project was completed, the first draft in 2000, and then later on another release in 2002. And it really caused me to see how much technology has a huge potential to influence sort of our basic findings in biology. And in a similar way, I really hope that sort of new development of technologies will really help us in sort of current mapping effort. But before we think of new technology, let's first understand what are the limitations of current mapping technologies. And there are sort of two aspects of the current mapping technology that I think are sort of still challenging. The first is simply just a matter of throughput. So I'm showing you here just a plot of a recent analysis of just the number of cells per study in different mapping technology over the last decade. So first of all, it's very encouraging, right? Every few years, there is a really strong growth, right? You see this sort of scales going from 100 to thousands to tens of thousands to 100,000. But still, it means that currently, the average number of cells per each one of those sort of, in a given paper that try to map the brain or any other organ is still relatively small, around 20,000 of those cells. And then another inherent challenge is the thickness, right? A lot of the mapping technology are still limited to relatively thin sections, right? Mostly 0.01 millimeters, right? Or 10 micrometers. Really small section that kind of limits your ability to really appreciate the complexity of a whole organ. And really what I would like to be, the world I would like to imagine is a world where we can take a centimeter cube, which is really the type of a biopsy, size relevant to a biopsy in a clinical study, or roughly the volume of the mouse brain. And that, as we heard many times, has roughly 100 million cells, right? So with current technology, it will take more than a year of just imaging time and to image all the 1,000 sections in that with the cost of more than $10 million. So it's doable, but it's definitely not something that an individual lab can undertake. So in my lab, we're trying to identify sort of new technology that will allow us to overcome those limitations. And let me sort of share with you kind of just our approach of how we approach this problem. So what I'm showing you here on the left side is a small, really tiny fraction of a mouse brain. And every dot represents one type of molecule in that brain, right? Those are sort of RNA molecules for the aficionado in the audience. And what do we do with this type of data, right? To use this data as an input to any sort of creating maps, you run it for some computational algorithms, right? And those algorithms inherently do two things. First, they perform some form of compression. All right, by compression, I mean that, there's a lot of information in this, even in this tiny image, there is a lot of information. So you really want to make sure that you get this essential information, right? You want to distill the essence from the noise and really represent the same amount of information with maybe less numbers, right? To get the sort of the signal, get the stuff that really counts. And only on those numbers, right? Only on those sort of reduced information, you do your label assignment and assign cell to type. Those are excitatory neurons, those are inhibitory neurons, sort of, etc. And let me delve a little bit more into this question of compression. And rather than neurons, let me start with sort of NBA players as an example. So if I wanted to look at NBA players, right, I can look at their height and I can look at their weight. And really, you can ask, do I really need to know both pieces of information? Do I really need to know their height and their weight? Or could I somehow summarize that only by saying that they have a specific size, right? And it makes sense, right? Because the height and weight are very correlated. So maybe we can compress that information and really describe NBA players only by their size, rather than displaying them both by their height or their weight. Now, if you go to NFL players, you will see that that becomes even more exciting, because here, right, if I summarize, right, it's not linear anymore, right? There's sort of, at some point, there is a limit to their height. But if I summarize their sort of relationship between height and weight, not only I can sort of reproduce it with only a single number, I can really just tell you based on their position on that curve, is it a linebacker, is it a quarterback, etc. Right? So knowing where you are on that line can really tell me a lot about what that player is, right? So I was able to summarize all that information with sort of compressed sense. right? So of course, finding that how to do that, right? Finding how to compress the information about player traits is really sort of a key computational task. Now, in the same way that we have NFL players, of course, we have neurons, right? So in neurons, we also try to do the same thing. We ask ourselves, can we find a way to sort of, rather than measure every small things about the neurons, rather than measure every different types of molecule in it and the different abundances, can we actually find out sort of stains? Can we develop new biochemical stains that will allow us to, that will show us the essence, will show us really the key information that we need in order to say what type of neuron it is and where it is, so we can build those maps with much sort of easier measurement, right? So really the idea is that different types of neurons have those different molecular attributes, and sort of we develop those algorithms to find those patterns, use those patterns that already exist in neurons to find those molecular signatures and allow us to develop dedicated biochemical stain to show us where they are. So really, if I go to this sort of approach, right, really the essence of what we're trying to do is to say, can we ahead of time perform that calculation, know what is in each one of type of neurons, and develop those specialized stains that we can stain those neurons, and really then on using all those stains together, right here in the middle, I'm showing you 24 of those dedicated stains. With those set of 24 stains, sort of staining the tissue kind of sequentially, one round after the other, right? We can then use that information to perform the mapping operation that we need. So this really helps us, right? Because rather than measuring those individual molecules, right, which is really sort of very kind of labor intensive and time consuming, will allow us to measure those aggregates that still capture all the information that we need to allow us to build those maps. So let me show you how it looks. So this is an example, right? So it is a mouse hemisphere, sort of half a mouse brain, kind of on the side. And you can see here, this is much bigger, right? So this is there are 50,000 neurons in this image, and the hippocampus that Hong-Wei showed you earlier is this sort of small region up here. And this is the same section that we stained multiple times using multiple stains. And then computationally, we combine all the information for those stains to sort of try to identify what are the different regions in the brain, where they are, what is happening there, and really use that information to sort of then create those specific maps, really giving every cell its own sort of specific identity. Right? So really in my lab, we're trying to build a technological solution to allow us to do this mapping effort better, really trying to combine instrumentation, algorithms, and sort of experiment to really create sort of scalable mapping efforts.
Wow, that was fantastic, Roy. That's really amazing innovation. I think I liked your example of the NFL players, where you really need to just see this one measurement of a combination of size and weight to be able to know what role that player has in the team. So thank you very much. I'll bring you back for the Q&A. So now it is my pleasure to introduce to you our last speaker, Dr. Jessica Lee. Jingyi Jessica Lee is an associate professor in the Department of Statistics. She is also has appointments in the departments of Biostatistics, Computational Medicine, and Human Genetics. And she's also a member of the Institute for Quantitative and Computational Biosciences. Prior to joining UCLA, Jessica obtained her Ph.D. at UC Berkeley in 2013 and her bachelor degree at Tsinghua University in China. Jessica and her students focus on developing statistical and computational methods to address important biomedical science questions and analyze large-scale genomics and health-related data. Jessica is the recipient of the Alfred Sloan Research Fellowship, the Johnson & Johnson Wisdom 2D Math Scholar Award, the NSF Career Award, the UCLA DGSOM Keck Foundation Junior Faculty Award, the MIT Technology Review 35 Innovators Under 35 Award, and most recently, the Harvard Radcliffe Fellowship. And so please join me in welcoming Dr. Jessica Lee.
Thank you very much for the introduction, Alex. So it's my pleasure to present this genomics technologies about mapping the brain, following the first two wonderful talks that introduce how we want to map the brain and what the techniques and challenges are. So following Dr. Roy Wollman's talk, I will focus more on the exact technologies and the computational challenges involved. So Mapping the brain is a very big challenge because we have a lot of cells in the brain tissue. So this is just an image showing that we can place a brain slice on a microscope to visualize the nerve fibers. But we are not satisfied for just seeing this image. We want to look into deeper details. So basically, in recent years, in the last decade, there has been international initiatives to map the brains. So US played the biggest role in this effort. So there's a US Brain Initiative and until 2027. And in Europe and in Japan, there are also efforts. So the common goal is to understand our brain much better. So the BRAIN Initiative, in the first part of it, or a part of this BRAIN Initiative is called the Cell Consensus Network, so short as BICCN. And the goal is to build an atlas of brain cells, including the questions, how many different brain cell types are there, in what proportions do they exist, how they are spatially arranged. To answer these questions, many technologies were used. So on the right is an image showing that we are interested in knowing the morphology of brain cells, the connectivity of brain cells, and the locations of brain cells. And at the molecular biology level, going into every cell, going to the nucleus of every cell, we want to measure how much each gene is expressed as mRNAs. So this is the molecule that transmits information from our DNA and converts the information into protein, because protein will play most of the roles in our cells. So gene expression is an important feature to measure in every cell. And chromatin accessibility is another molecular biology feature. People call this as openness. So because our DNA is open in some parts, you can see that the naked DNA is called open, while if the DNA is tightly wrapped around some proteins, it's called closed. So open and closed have different implications in our nucleus. And the third is DNA methylation. In the DNA or on the DNA, do we have some sites methylated? If so, what does it mean? So this is also a known interesting feature of our molecular biology. So gene expression, chromatin accessibility, DNA methylation will be measured by technologies. Besides these molecular biology features, we also want to measure the electrophysiological features of neuron cells. So together, the BRAIN Initiative Cell Consensus Network have used these cutting-edge technologies, including single-cell RNA sequencing, MERFISH, patch-seq, single-nucleus DNA methylation sequencing, single-cell ataxy, which I'll briefly introduce in the next few slides. And currently, the focus has been the brain's motor cortex because it plays a very important role in our brain. So following those technologies, we will get data like this. So this shows a brain tissue slice, and the colors indicate different genes expression. So one color for one gene. So this will give us some pattern about how these genes are expressed in the tissue slice. And also, we know the cell's shapes, their morphological features. So a question will be, how do we connect gene expression levels to the cell's shapes? And what do they mean? What do the relationships mean? And finally, how the cells are connected. Are they connected within the cortex, or do they travel from cortex to brainstem? So include for the technologies, single-cell RNA sequencing has become very popular. So given the brain tissue, we want to first isolate the cells into individual ones, and using the sequencing technology to measure the RNA molecules in each cell. And after sequencing, we will know, in each cell, which genes are highly expressed at what level. So this data will allow us to divide our cells into clusters that share similar gene expression. And in each cluster, we can tell which genes are more highly expressed than in other clusters. And this way we can understand the cells better, and also to find cell types. And MERFISH provides additional information about the spatial information of cells. So single-cell RNA-seq dissociate cells, so cells' spatial locations are removed, no longer available. But MERFISH is one of the so-called spatial technologies which allow us to record the locations of the cells, and each dot, a different color, means a different gene. So we can see where the genes are expressed. And so this is like when we see the whole tissue slice, we will see the patterns of different genes. And PATCH-seq provides additional information to single-cell RNA-seq by measuring the electrophysiological properties of cells for neuron cells. So this will allow us to do the morphological reconstruction of the cells. So besides just having the gene expression levels in individual cells, we collected more data, more features of the neuron cells, so allowing us to understand the cells better. And single-cell nucleus DNA methylation sequencing is a complementary approach to the single-cell RNA-seq, which measures the collection of RNAs, referred to as transcriptome. So in addition to RNA information, this sequencing technology provides where the DNA is methylated in every cell. And this allows us to understand how in each cell or cell type, the highly expressed genes are regulated by which part of our genome, because the majority of our genome are not genes, but they have their functions. So understanding their functions will require additional information, such as DNA methylation. And finally, we have the single-cell ATAC-seq technology. It is also complementary to single-cell RNA-seq and DNA methylation. It measures where our DNA is naked. It's not tightly wrapped around proteins. So knowing this information will also allow us to understand how the active genes are regulated in each cell type. So using single-cell RNA-seq and ATAC-seq data, we will be able to project our cells to this two-dimensional space where every dot is a cell, and we will see the cell clusters, which may indicate cell types. So this will allow us to identify new cell types or intermediate cells that do not clearly belong to one cell type. So we can discover those information from these data, which give us hundreds to thousands to millions of cells in one experiment. So given those cutting-edge technologies, we are still very far away from understanding the brains well. So this is a quote by Dr. Hongwei Dong, who is the director of the Allen Institute for Brain Science, which this institute has invested a lot into collecting data to understand human brain. So Dr. Dong said, we wanted to be able to see every gene that expressed in every cell at the same time. However, we're still very far away from this goal. The challenges include experimental and computational scalability. So mouse brain has more than 100 million cells, 2/3 of which are neurons. Human brain is 3 orders of magnitude larger, with more than 170 billion cells, of which half are neurons. So how can our technology effectively measure so many cells in a reasonable time? And also, how can our computers be powerful enough to analyze such massive data sets? And also, data quality is another thing we must bear in mind. If we increase the throughput of our technology so we can measure more things at the same time, we usually lose data quality. How can we balance the two? And also computationally, because I'm a computational person, the computational complexity is something we must bear in mind. So if we measure different features using different technologies, how can we integrate data in a meaningful way? And what scientific questions can be answered from these data, these new technologies? And how do we ensure the validity of data analysis? So basically, these will call for new methods, new computational methods to address these challenges. So I want to say that one challenge I can show here is that every tissue is different. The brain tissues are structured while cancer tissues are not structured. So therefore, we must bear this in mind. So to help with this tool development and computational analysis in this field, my lab developed a simulator which will allow us to generate some synthetic in silico data so we can use those data and our specified ground truth to help benchmark computational tools. So thank you all for your attention, and I welcome questions.
Thank you so much, Dr. Lee. This is really exciting. I'd like to I'd like to welcome back here now Dr. Wollman and Dr. Dong to join us for the conversation, and there's some interesting questions here. I have to start off. I mean, all three of you provided very different approaches to mapping the brain, right? So Dr. Dong started off with microscopy largely and looking at the neural shapes and the connections. Dr. Wollman described a ingenious technology to make a single measurement to identify a number of different neurons and therefore on a large scale. And Dr. Lee then said, we need to have all of these different types of measurements that go into much more detail. And so what is the right answer? Are you competing, or is there complementarity? Is there competition? Maybe I'll ask Hong-Wei first to address that, because he's been in this space for longest. Can you unmute yourself? Sorry. Can you unmute?
Oh, sorry. Okay, unmute. Yes, absolutely. This is complementary technologies. Actually, we are collaborating with each other, and with Roy, we are trying to collaborate, because just like what Jessica already present, in order to understand the brain, we need an approach from different angles. My angle is from anatomy, look at the cells, their neural connections. From Roy's angle, that is really understand the genomics, and Jessica, that is use computational tools, put everything together. So absolutely, we need all these technologies together. Jessica mentioned that BICCN, that is an enormous collaboration, one of these groups, and to work together with many other institutes, many other collaborators. Hopefully, we can put all the effort together, or we can generate a much more larger data set and also understand more deeply for the scientific questions.
Yeah, that sounds great. It's going to be very exciting. So let me turn to some questions. Actually, there's a question that was asked by an attendee, Charles Michul. Based on all the failed clinical trials in Alzheimer's disease, it's apparent that treatment needs to start decades before the disease becomes behaviorally apparent. How can the brain connectome be applied to humans decades before there's cognitive impairment? And how do you see this becoming a diagnostic? Maybe Hong-Wei, I'll ask you again on this one.
Yeah, this is a great question, and also it's a really great challenge for the entire field. So what we're trying to do here is use a mouse model, because the mouse model, you can control their at which stage. Use a mouse model of the AD, we can look at the earlier stage. What is the earliest stage? Their neural circuitry changes, especially for the neuromorphology changes, like individual neurons, or whether they are dendritic changes. So we can try to look at a really earlier stage, understand in that stage what gene changed, and then use this information to predict what happened in the human brain. So that is still a really challenge at this moment. And hopefully we can get some more information, not only from my groups, but also like across the entire world. So many groups, they are trying to answer these questions. They produce more mouse models, use all these different technologies. And Roy and Jessica, probably you can add something more.
Sure, let me chime in here. So I think what is amazing sometimes is that there are things that our body is doing that we're not even aware of, right? So only recently we found out that we can find which sort of cells that are dying in your body are sort of excreting some of their contact back to the bloodstream. And simply by looking at your bloodstream, scientists can now identify which tissue is now sort of going over a different level of deterioration. So there's things that we still just don't understand yet enough about how the system works. And definitely improving our basic understanding of things is key to any development of technologies. So if you're asking me, can I use the exact technology that I'm now developing on the human brain? No. I can't sort of take a human brain and slice it. That of course will not, right? That'll be unethical. But can I actually do things on a mouse and learn from them? And then from that basic biology, things will emerge that will be applicable to humans? Definitely so.
That's great. So maybe I have another question here. That ties into what Jessica was talking about. So how important are these advances in these sequencing technologies for mapping the brain? Is it not just all about microscopy? Why do you need to have sequencing technologies to mapping the brain, Jessica?
I think at a high level, we want to go to the molecular biology to understand what might be the genes that cause certain morphological changes in the brain tissue, because knowing so is a way for us to develop possible drugs if we can target a certain gene's protein in some way. So going to the molecular biology will allow us to understand from the mechanistic level about what changes might have happened before the disease occurred. So I think this is how we want to connect I should say macro scale morphology to the micro scale molecular biology. So this is how the biomedical research goes since maybe 50 years ago.
That's great. Yeah. And then going back to the imaging that Roy was talking about, how is that imaging that you describe used to make maps? Is it similar to the way people do in the clinic, do MRI or CAT scans?
Not exactly, no. So, right, MRI and CAT scan were developed exactly to overcome the challenge that we can sort of introduce stains and dyes to a tissue, right? But when we work on sort of our animals, when we work on mice, right, it's a different game, right? So we can take our animals, we can look at them, and we apply a lot of different stains. So really what we're measuring is sort of the staining patterns, right? We stain those dyes either with direct color or a lot of time use fluorescent colors, right? And use those sort of the image, right? Our cameras then sort of just capture the patterns of staining, the pattern of fluorescence in each sort of region of the brain, and those are being used to sort of reconstruct those maps. So no, those are qualitatively different from sort of MRI, right? That are really trying to solve the problem of how can you create maps on a living organism. The challenge, of course, with MRI is that the resolution is really sort of in the millimeter scale, right? Not enough to really go to sort of cellular level, and definitely not providing molecular information.
Will it be possible to maybe have some probes in the future that that allow you to do MRI and still have some kind of measurement of specific cell types? Because I know that there's contrast agents that people use for cancer screening and CAT scans. Might you be able to, can one imagine that there might be stains that you develop also for particular neural types that can be imaged in a non-invasive manner?
Yeah, I can definitely imagine things like that will happen. I think one of the biggest challenge will be a matter of scale, right? You can develop a stain for a given neuron, and if you can have a key indication and that specific neuron type is important for specific diseases, that will be fantastic. That will really be a huge benefit for sort of human health, depending, of course, on the details. But if you're really trying to build sort of large-scale maps, right, that will not give you the sort of scale that you need, what you can do with sort of direct staining of tissues.
So this will be useful when you have a map, right? When you have the map, and then you don't need to look at every neuron, but you can then stain for the one specific one that might be implicated in Alzheimer's disease.
Yes, exactly.
Yeah. All right, and here's another question, and I think it goes to Jessica's point that there are differences and similarities between different organs, the brain and other organs, and people are mapping how cells are arranged. And how different are are they in applying genomic technologies and statistics? From your point of view, what's special about the brain in that regard?
That's a very good question. I think given that we have some knowledge about the brain structures, and I think these knowledges should definitely be used. And I think then the question is, what new information can we gain from the new technologies, the genomics technologies, in addition to what we know? So I think there are a lot of open questions right here because the technologies are still very new and the many data were recently available. So what information can we extract from this data? I think that's a very big research opportunity.
Yeah.
Alex, if I may add to that. So one of the things that attracted me to the brain is really the diversity, right? A lot of organs have the same idea and you can apply the same technologies, but sort of before the brain, we looked at the sort of a cornea. And the cornea is also a really important organ, but not very complicated one, right? Whereas if you move to the brain, right, you have such an immense diversity of types of neurons and their molecular patterns, right? There is something really sort of exciting about charting such as sort of, such an organ. So the technology is similar, but I think brain does have sort of a qualitative difference in its complexity.
Yeah, the complexity really also is because there are connections. So if we look at the individual neurons, each individual neurons, their connection can be, they can be next to each other, but their connectivity can be enormously different. Like Jessica mentioned that BICCM project for the motor cortex, we mapped, let's see, hundreds of neurons in the same region. You can see this tremendous diversity of their connectivity. Even they expressed the same genes, but their connectivity can be still really different.
Yeah. So Hong-Wei, earlier you talked about the comparison between artificial intelligence networks, neural networks, artificial neural networks, and our natural neural networks in the brain. So and you talked about how the natural ones are so much more complex. So by studying them and understanding them, is that going to make will we be able to make AI, artificial intelligence, more powerful?
Yes, absolutely. That's what I hope for. And I think is many people hope that will happen in the future because the current artificial neural network that is really just based on the really fundamentally, the most fundamental neural connections. And as what I have showed, actually I showed that on purposely, is I want people to understand the actual neural networks so much more complex. I just showed the hard wiring. I even didn't mention like their connections. They have excitatory part, which part is the inhibitory part. They have recurrent connections, just tremendously complex. You know, I hope in the future, once we can build up like a more like complex neural networks based on real actual neural networks, that will be like make the real intelligent artificial intelligence even more intelligent.
Yeah. Jessica, you're in the statistics department, so you know all about statistical learning. What do you think about these how can one generate better deep learning algorithms based on the insights from the brain? Do you have an idea about that?
That's a very good question. I think we are still very far away from there. First of all, the number of neurons in those artificial neural networks are not in the same size as the actual number of neurons. That's the first thing. So therefore, any connectivities we learn from actual neurons will not be directly translatable to the neural network, artificial neural network. However, maybe I think something related to the connectivities at the cell type level may give us some indications. So yeah, we're not as ambitious as to make the real brain, but if we can borrow some information from there, that may be helpful. Yeah. And also I think actually adding human logic into the neural network architecture will be, well, at least people are doing this to make the neural network, artificial neural network more interpretable.
Yeah. I think Roy is also thinking about AI and how it might in some way mimic the human brain more. Roy,
yeah,
what are your thoughts?
There's definitely a lot of sort of kind of in really interesting synergy that goes sort of both direction, right? We use a lot of AI actually as a tool in our work, right? To sort of allow us to help interpret some of this data. And then what we learn from that could potentially make sort of better AI. And who knows what happened if you walk in that cycle for a few iterations, right? I mean, the future is definitely exciting.
But for now, I guess we can all be reassured that our brains are a lot more powerful than what AI is able to provide us. So we're not going to be facing unemployment anytime soon. So far, the creativity of the research will keep us in place. So,
yeah.
Well, with that, I really thank you for those presentations. We really enjoyed them, and the conversation was really exciting. And I think it's giving us a perspective of both the importance and the challenges of mapping the brain. So I think there's some other questions in the chat we weren't able to get to, but I hope that all of you who are listening had enjoyed the presentations and the discussion, and we hope to see you at future events. This was just one of several ways that you can engage with us at the Institute for Quantitative and Computational Biosciences, and so feel free to look at our website and sign up for email newsletters. And so let me thank the presenters again for wonderful presentations and discussion. And so I leave you for a nice evening at this point. Thank you very much.
Thank you very much.
Thank you, Alex, for this opportunity.
Thank you.